important topics in biochemistry. This is part of a video series of small lectures that I'm going to go over some of the important concepts, 1 out of 5, 1 out of 15. We'll cover glycolysis, the pyruvate dehydrogenase complex, lactate dehydrogenase, and some clinical correlations as well as some rule of thumbs for you to know. All right. I put here that I have questions on other slides, but that's actually for a different lecture that I have. So the first step in glycolysis is hexokinase adding a phosphate to glucose. Remember, glycolysis takes place in the cytoplasm. And in order for glucose to not leave back into the extracellular environment, it needs to be phosphorylated because that gives it a charge, and charged molecules cannot cross the cell membrane that easily, right? And that's what hexokinase does. And the hexose is because it's a 6-carbon sugar, and the kinase is because it adds a phosphate. And then glucose 6-phosphate is because when you count from the most oxidized carbon, this aldehyde right here, you go 1, 2, 3, 4, 5, 6, and you see a phosphate there, so glucose 6-phosphate, right? Now, the next step is phosphoglucose isomerase. An isomerase simply just rearranges a molecule. In this case, glucose gets rearranged into fructose 6-phosphate. It's important to know for your future coursework in biochemistry that an aldose is a different type of sugar than a ketose because of the aldehyde there and the ketone there. Ketone is simply a carbonyl carbon with 2 adjacent carbons on either side, right? Versus aldehyde, which is simply half ketone, but instead of the other CH3, you have a hydrogen, right? And the next step, which is the most important step, is going to be the rate limiting step, meaning that this step is irreversible, all right? That means that it's irreversible. That means that it cannot go back, obviously. But the important thing to know is that this is the rate limiting step because it's a slow step. That means that all of these reactions relative to this one are much faster, all right? And the enzyme is known as phosphofructokinase. Phospho for the phosphate being transferred onto fructose, kinase being an enzyme that adds a phosphate. So phosphofructose kinase, all right? It adds a phosphate to fructose with a phosphate. And it's also referred to as PFK. And it creates something known as fructose 1,6-bisphosphate. Now let's see how this name emerges, all right? Where's our most oxidized carbon? We can say here, right? So we go 1, 2, 3, 4, 5, 6, right? 1 for 1 and 6 for the other carbon, the other phosphate, so it becomes 1,6-bisphosphate. So always look for the one that's most oxidized. Rule of thumb, something that has double bonds is probably more oxidized than something that doesn't have double bonds. Actually, it's always. The more double bonds, the more oxidized. The more oxygens, the more oxidized. All right. But if you're comparing two molecules with both oxygens on them, the one that has double bonds is the one that's oxidized. The one that has hydrogens is the one that's reduced. And we'll look at that later when we talk about the lactate dehydrogenase reaction. All right. Now, fructose 1,6-bisphosphate gets cleaved by aldolase. Here it says fructose bisphosphate aldolase. Aldolase is usually the only name you'll hear. And it generates dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde is important in fatty acid metabolism. It is derived from glycerol, which is basically just these 3 carbons, but you have an OH here, OH here, OH here. That's the only difference. OH, 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 instead of double bond oxygen phosphate or whatever. But that's why it's called glyceraldehyde 3-phosphate, because the glyc is glycerol derived, all right? And then you have dihydroxyacetone phosphate. To be quite frank with you, I don't know why this is called dihydroxyacetone phosphate, because you have an acetone, 1, 2, 3, right? You have one hydroxyl group and you have a phosphate. That makes sense, but I don't see another hydroxyl group. It's either that this picture has a typo or that there's some weird exception going on here, all right? All right, so the next step is basically this glyceraldehyde 3-phosphate is going to be oxidized by glycerolide 3-phosphate dehydrogenase. Anytime you see dehydrogenase, always think oxidation reaction. And NAD plus is going to be reduced to NADH. NADH has reducing power and it's going to be used to reduce other things as well. In the future, you're going to talk about it in the electron transport chain, as well as the lactate dehydrogenase converting pyruvate into lactate, all right? So glycerolide 3-phosphate is going to generate 1,3-bisphosphoglycerate. This makes sense because you have 2 phosphate and you can count them 1, 2, 3, and that tells you exactly why it's named that way. Now, phosphoglycerate kinase is going to generate 3-phosphoglycerate by losing a phosphate, and this is going to be different from the previous steps. If you noticed, in these reactions, we invested ATP. So ATP was used, but here, ATP is being made and is being taken away from a substrate. And that's why this is known as substrate level phosphorylation. It's one of the ways ATP is made outside of the electron transport chain, all right? Now, you have 3-phosphoglycerate because we lost the phosphate. Now phosphoglycerate mutates. Mutate sounds like what? Mutate, right? So what do you think it's going to do? It's going to mutate something. It's going to move something around. It's going to alter its typical appearance. So what happens here is this phosphate gets swapped with this hydroxyl group. Simple as that. All right. So you get, instead of 3-phosphoglycerate, the position at 3, 1, 2, 3, you get 2, 1, 2. All right. And these endings can tell you something about the chemical structure as well, and you'll see it very common. Something that ends in 8 has a negative charge on it. If it ends, little tricks like that are going to basically save you from memorizing a lot of information sometimes. The 2-phosphoglycerate is going to be converted into phosphoenolpyruvate by enolase, and water will be lost. So this is a dehydration reaction. Water is lost. And where is that water coming from? If you look closely here, you'll notice that this hydroxyl group disappears and this hydrogen also disappears. And water is what? H, 2, 1, 2, and then O, right? And then this bond that was between this carbon and this hydrogen is going to reform right here. And that's why you see a double bond right here. And I'm going to zoom in so you can see it closer. So you lose water, right? Sorry, you lose water, this OH, and that this OH and this proton, and the bond that's left is going to basically reform here and create this double bond right here. And that's what the enzyme does. And this is a dehydration reaction. Why? Because you lose water. You generated phosphoenolpyruvate, all right? Why is it called phosphoenolpyruvate? Why do you think it's called phosphoenolpyruvate? It's important to try to understand why things are named the way they are, because you don't have to memorize so much once again. Now, phosphoenolpyruvate is important also in substrate level phosphorylation because it's going to phosphorylate ADP, adenosine diphosphate, to ATP. And this is another example of substrate level phosphorylation. And by losing that phosphate, what do you think is going to happen? It basically becomes pyruvate. The phosphoenolpyruvate via pyruvate kinase gets converted into pyruvate, and ADP gets phosphorylated in the process from the phosphate of phosphoenolpyruvate. This is known as substrate level phosphorylation. Thank you for listening. If you have any questions, please send them in.